Today, I'll be reviewing a popular science article from 2023 focusing on epigenetics and its impacts on cancer research. When we think of cancer, we first think of mutations in our genome causing tumors and metastases. But there have also been a number of cases in which scientists couldn't seem to find a genetic mutation. This is because our epigenome can also contribute to cancer occurrence. While our genome is the easily identifiable DNA material present in all of our cells, our epigenome is the dynamic and hard to pinpoint group of factors that control and regulate the expression of our genes. Factors such as non-coding RNAs and transposable elements are within our epigenome. The article explores this idea, also referencing the fact that there is a higher chance of cancer in older generations, not only because of genetic mutations over time, but because of the loss of epigenetic information contributing to the mutation phenotypes. Mathematicians have been using what's called stochasticity, or the unpredictability due to randomness, to better understand the phenotypic plasticity of cancer. In doing so, they hope to give us a leg up in our journey to creating cancer therapeutics and treatments. Because of the relatively undefined nature of the epigenome, I found it difficult at first to comprehend the complex ways that it interacts. However, after studying transposable elements and non-coding RNAs in the GEN2052 unit, I was able to break it down and understand some of the moving parts. Now I recognize that epigenetics is the linking factor between cancer and stochasticity. The article's final statement addresses this evolution of biological research and mathematics merging. It begs the question of how the basic factors we learn about in GEN2052 will grow to more strongly predict gene function, relating to unit outcomes 1 and 2. With bioinformatics and large-scale data accumulation within the genetics research world, predicting impacts of gene alteration can become increasingly accurate. Tools such as NCBI BLAST and genome browsers, like within Outcomes 1 and 6, also relate to this, through the comparison of genetic and epigenetic information. Researching epigenetics and the non-coding genome has revealed to me that although cheaper and more accurate methods of sequencing, such as Illumina and third-generation technologies, exist, there are still many more developments to be made. The epigenome keeps the genome slightly unpredictable, so perhaps techniques of a wide-scale sequencing of the epigenome on an individual level is our next big step.